We have now discussed a few substrates, that is creatine phosphate. Then we went to glucose and glycogen. Now we're going to talk about how do we use lipids as our fats for fuel. So first thing you need is lipolysis. Just like we say glycolysis, always break the word down. Olysis means to break down, and then lipids. So this is lipolysis is the breakdown of lipids. Lipids are stored in adipose cells as triglycerides. A triglyceride is made up of a glycerol and 3 fatty acids. So if you're ever having trouble with the names, just break the names apart and it'll help you to figure out exactly what that is occurring or what is it made up of. So lipolysis is the process which cleaves the fatty acids from the glycerol. The free fatty acids and glycerol now enter the bloodstream. So the problem with lipids, then the last point right there, is lipids do provide a lot of ATP. There's a lot of energy that comes from lipids. However, the problem is that we've already talked about multiple steps right now just to get to the fatty acids and the glycerol into the bloodstream. So there's a lot of room for error with trying to break lipids down for fuel. To become really efficient at using lipids for fuel is a training adaptation. So those who are more trained will be able to use more fats just because there's a lot of processes that go into this. So the more training, the better off you are at being able to use the lipids. Because you can see first you have to cleave the lipids free, then break them into then the triglycerides, and then down to the glycerol and the free fatty acids. Then you have to get them into the blood. And once they're in the blood, then you actually have to ship them around to where they need to go to. Okay, so once you're in the blood, then you got to get it to the site. And so you've seen these multiple steps occurring just to be able to use just one free fatty acid. Once the free fatty acids get to the cell, they're able to get into the cell, but then they need to carry it into the mitochondria. So now we're adding more steps. So those free fatty acids must enter in the mitochondria. However, it says long-chain free fatty acids need carnitine to transport them into the mitochondria. So free fatty acids could get into the cell, but they can't get into the mitochondria. So you need carnitine. Remember that because I always will ask about carnitine on exams. But you need carnitine to be able to transport the free fatty acid into the mitochondria itself. So that's another step. Okay, there's a lot of enzymes that go along with this. And so just because you get the mitochondria outside of or the free fatty acids outside the mitochondria ready to be taken in, well, you need carnitine. But then you need the enzymes that catalyze that reaction and get it in the mitochondria. So there's a lot of steps, a lot of chances for failure. But then again, this is why training becomes so important. The more you trained, and I know I brought this up briefly in previous, but we talk about oxidative system, and the more you train, the more enzymes you get, the more pathways and so forth. I've said that maybe 1 or 2 times already. Well, training does that. So the more you train, the more enzymes are developed, the more pathways are developed, and the better off you are able to get these processes to work, that is, getting the free fatty acids into the mitochondria, then to go through what we'll talk about here in a sec is beta oxidation. So remember carnitine and the importance of that. To get this whole process started, you need, it says, entering into the cell results in the net loss of 2 ATP. So 2 ATP kick off, you need to basically invest 2 ATP to get into the process of beta oxidation, which is point 7, which we'll talk about here in a sec. Okay, so remember, just to get to the mitochondria, there's many steps, multiple steps, a lot of chances for failure. So that's why training becomes so important, it allows for all these processes to occur faster and a lot more efficiently. So that was lipolysis. Now we'll talk about beta oxidation. Beta oxidation occurs in the mitochondria. And beta oxidation is when free fatty acids are broken down into acetyl-CoAs. And acetyl-CoA, I didn't say this when we're talking about Krebs cycle, but an acetyl-CoA is a 2-carbon chain. Remember how we talked about glucose 6 carbon, then pyruvates are 3 carbon. So I also said the tricarboxylic acid cycle where you give off CO2. Well, when pyruvate is broken into acetyl-CoA, we go from a 3 carbon to a 2 carbon. 3 carbon being pyruvate, 2 carbon being acetyl-CoA. So you lose 1 carbon, you give off a CO2 at that point, and that's when one of the CO2s is given off. So remember, acetyl-CoA is a 2 carbon molecule. And just know that beta oxidation is the process of taking free fatty acids, which a free fatty acid is just a long chain of carbons. Multiple carbons. And so what beta oxidation is doing is taking this long chain of carbons and cleaving off an acetyl-CoA group throughout the entire chain. That is, beta oxidation is the process of cleaving off 2 carbons at a time. 2 carbons at a time. 2 carbons at a time. And that's what we'll get into next. So beta oxidation, aka fatty acid oxidation. It says free fatty acids are long chains of carbons, usually 6 to 24 carbons in length. 18 carbon stearic acid is the most common free fatty acids found in humans. It says free fatty acid is cut into 2 carbon units, which is an acetyl-CoA. Each cut yields 1 NADH and 1 FADH. So every time acetyl-CoA is cut and given off, an NADH and the FADH are given off as well. So this is, now you can see, going back to what we talked about with the Krebs cycle and ETS, we know acetyl-CoA, the process going through Krebs into then the ETS, gives off ATP, the whole process there. But then we also have here NADH and the FADH. So it's even more ATP. So if you could kind of just think back to what we've talked about so far, all 3 of these molecules in the ATP process, we're starting to get a lot more ATP from just one fatty acid. It says each 2 carbon, that is each acetyl-CoA, enters into the Krebs cycle where 3 NADH, FADH, and GPT are obtained. This yields about 10 ATP via the ETS. Okay, so just to kind of recap on here, beta oxidation is taking this long chain of carbons, cleaving off 2 carbons at a time, which is an acetyl-CoA. And each time there is a cut, that is each time acetyl-CoA is given off, there is then an NADH and FADH given off as well. And you can see right here, just think of these are the cuts right here. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 carbons. So how many acetyl-CoAs are going to be given off? So 1, 2, there's 1 acetyl-CoA. Here's an NADH, FADH right here given off well. Each line represents an NADH and FADH. There's 1 acetyl-CoA. 1, 2 there again. There's another acetyl-CoA. And when I say 1, 2, I'm talking about counting the carbons there. NADH and FADH. Here's another acetyl-CoA. There's a cut. Acetyl-CoA, cut. Acetyl-CoA, cut. And acetyl-CoA. So I'm trying to figure out how many ATPs given off. First, you count up the amount of acetyl-CoAs. So if there's 12 carbons, then you have 1, 2, 3, 4, 5, 6. There's 6 acetyl-CoAs. And how much NADH and FADH? You count the cuts. 1, 2, 3, 4, 5. 5 of each NADH and FADH given off. There's always going to be 1 less NADH and FADH than acetyl-CoAs, just because when you get to the final 2 acetyl-CoAs, there's only 1 cut, so there's only going to be 1 less cut than the amount of acetyl-CoAs. Point being on here, there's a lot of ATP given off from 1 fatty acid. So kind of let's go through how much ATP, and I'm not know these numbers generally speaking. There's not going to be a specific on the test, more or less, but just know if there is something on the exam, I should say, then it's going to be something real that you're able to figure out without having to memorize the actual numbers. Okay, but it should be simple. If I give you amount of carbons, can you come up with how much roughly ATP? So here we'll say we got stearic acid, so an 18 carbon. Well, always just take the number of carbons, divide by 2, and that gives you the amount of acetyl-CoAs. So 9 acetyl-CoAs. Remember, there's always one less NADH and FADH than acetyl-CoA. So that means this carbon right here, there's going to be 8 cuts. So there's 8 NADH and FADH. Times that by 2.5, times that by 1.5, gives you 32 ATP. Then you got the 9 acetyl-CoAs and the Krebs. So each acetyl-CoA gives you 3 NADH, 1 FADH, and 1 ATP. Remember, GTP and ATP are the same thing. So you do the math, 90. Remember, you have to invest 2 ATP, get the whole process started, so you minus 2. So you do the math, 120 ATP roughly from one fatty acid or free fatty acid that's 18 carbons in length. Now, remember, with a triglyceride, it's a glycerol plus 3 fatty acids. So from one triglyceride, this is just from one free fatty acid. So you remember, you got to times this by 3 because we deal with the triglyceride, which is 3 fatty acids. So roughly we're looking at 360 ATP from one triglyceride. And glycerol itself could enter into the glycolytic pathway and gives out, I believe, about 18 ATP because it enters into the glycolytic pathway pretty far down in the pathway itself. So you get roughly 18, I believe, from glycerol. So from one triglyceride, we're getting, a few hundred ATP as opposed to 30-some from glycolysis going into the oxidative system, 2 to 3 from glycolysis alone, and 1 from the immediate system. So fats do provide a vastly more energy than any other fuel source. And I should finish up with, remember, fats per gram, they carry 9 kcals per gram. For fats, carbohydrates, and protein provide 4 kcals per gram. So fat really is the energy. It's the preferred storage of energy for the body. We'll see later on, carbohydrates are preferred fuel source during exercise for the body, but we prefer to store energy as fat because of the density of it and just how much energy is contained within the density of fat. You got proteins. This is ideally, you never want to be in a place where you're using high amounts of protein as fuel. But what proteins are doing for the most part is they're being broken down into the amino acids. Then the body's using the amino acids to form up whatever molecule it needs, wherever it needs to be at. And it's another part too, where once we say the word deaminated. Okay, number 2, it says the amino acids must then be deaminated. Deamination means, amino acid, what defines them is the amine group, which is the N group on the molecule itself. That's what makes it an amino acid. And so we say deaminated, it's getting the NH, you see right here the NH3 removed. So problem when you're breaking down proteins is you're giving off ammonia as well. So this is another place where ammonia is being produced along with we talked about the immediate system. This is another place as well. So it's not good. You see 0.3, the NH3 being built up in the blood. It says the deaminated amino acids are used to make products which can enter into the Krebs cycle. Can enter at several points starting with pyruvate. It says some deaminated amino acids can be used in gluconeogenesis. I'll define that here in a bit. Some deaminated amino acids are used in the production of oxaloacetate. And you can see right here, all these different proteins, amino acids are being broken down into all these intermediates of the Krebs cycle. Okay, so I already talked about this, but this is another place where oxaloacetate, this is, remember, it's needed to form with acetyl-CoA to get the Krebs cycle going and started. Well, a lot of times when you run out of carbohydrates, you'll turn to start breaking down proteins and get the amino acids broken apart to then use to form oxaloacetate. That's usually when you're severely glycogen depleted, and that's never a good time to be in. But nonetheless, this is how we use proteins. Is we for the most part, when we're using protein for fuel, what we're doing is we're breaking proteins down into the amino acids to be used to build up at some place within the metabolic processes. You see right here within the Krebs cycle to be used to form all the different intermediates. This is always generally happen at all times. And when you're looking at proteins, there's always a relationship between breaking down versus building up. And there's a lot of proteins they do turn over quite frequently. That's not the problem. The problem is when you're having severe catabolism of your amino acids into form these intermediates. You never want to be at a point where breakdown is greater than building up. That's never a place where you want to be, is when you're breaking down far faster and far greater than you're building up. Okay, it said define gluconeogenesis, and this is defined as the manufacturing of glucose from a non-carbohydrate source. Just break the word apart. Genesis, the formation, the beginning of, neo, new, and then glucose. So the formation, new glucose. And it's coming from a non-carbohydrate source. So lactate, pyruvate, glycerol, amino acids can all be used to make new glucose. This occurs primarily in the liver, but can occur in resting skeletal muscle as well. It does require, the problem is when you get to this point, it does take a lot of ATP to just even get the new glucose. See, it says process requires as many as 4 ATP. So it's a costly process trying to form new glucose from these non-carbohydrate sources. It says fast muscles have a greater gluconeogenic capacity than slow muscles. Hence why we always say type 2 fibers are glycolytic. Okay, that's one defining terms of glycolytic, is be able to form new glucose within the tissue itself. Okay, just a little rehash on oxaloacetate. I know I've talked about it, but I can't stress this enough how important it is metabolically. It says fat utilizations depend upon OAA, short for oxaloacetate. OAA can only be made from glucose or amino acids. And it's done so, you can see right here, oxaloacetate, or you can see pyruvate, which if we have pyruvate, and ideally that's coming from glucose, that's going to form oxaloacetate. Or you can see, you can break down amino acids into the individual ones to give us pyruvate, or broken down into the individual ones to give us oxaloacetate. But it says fat utilizations depend upon OAA. You can see the acetyl-CoA right there on the right. Well, we get the acetyl-CoAs from the breakdown of free fatty acids. We just talked about that. And so remember, in order for Krebs cycle to get going, you need oxaloacetate and acetyl-CoA reacting with one another to give us then citrate. And so fat is dependent because you're not going to burn fat, that is the acetyl-CoAs coming from free fatty acids, unless you have oxaloacetate. The reality is oxaloacetate is always going to be there. It's just a matter of, is it coming from glucose or from a protein? Ideally, is you want to have enough glycogen and glucose available to supply the oxaloacetate to keep from burning up the amino acids in your protein for fuel. That's why I say fat must burn in the presence of carbohydrate, or you hear sometimes fat burns in the carbohydrate flame, meaning if you want to be efficient at burning fat, make sure you have enough carbohydrates. Okay, because the last thing you want to do is get to point 4. No carbohydrates present, fat utilization causes self-cannibalism. So if you want to get good at using fats, well, one, train and exercise and get the metabolic processes built up, but make sure you have enough carbohydrates in your diet as well. The carbohydrates will supply the oxaloacetate demands. The acetyl-CoA come from fats, and that will help out with metabolism without having to turn to the amino acids for fuel and for oxaloacetate regeneration. Okay, lactic acid, lactate formation. I've already talked a little bit about it, but just to kind of go through it again, it says lactase formed by adding an extra hydrogen to pyruvate. It says the hydrogen comes from the NADH, which is formed during glycolysis, upstream from pyruvate formation. This helps to keep glycolysis moving forward as fast as possible by keeping NAD high. So lactate, remember, is formed essentially when glycolysis is working at too fast a rate. And so it's formed because when glycolysis is working at a super fast rate, what's happening? Well, remember, the products of glycolysis are pyruvate, ATP, and NADH. And so glycolysis are working too fast. Well, so if it's working faster than the oxidative system can handle it, there's going to be a buildup. There's going to be a damming of the river. And so when that damming occurs, what is actually happening? Well, you're having a buildup of pyruvate, a buildup of NADH. And so the cell needs to clear everything out to make sure everything keeps working at a proper function. So what will happen is lactate dehydrogenase will then, the enzyme that will take the hydrogen from NADH, put it on the pyruvate, forming then lactate. At that point, lactate will get cleared out of the cell and it could go where it needs to go to. Primarily if type 1 fibers. Okay, lactate's a huge energy supply for type 1 fibers, primarily the heart. The heart has enzymes that can take lactate and turn it back into pyruvate. Your type 2 fiber is not able to take lactate back into pyruvate, but your type 1 fibers, primarily the heart specifically, has those enzymes that could convert lactate back into pyruvate, and then from there pyruvate could go through the pathways in the Krebs cycle and so forth. So primarily what lactate would like to do is be oxidized for fuel. So what lactate's doing is, one, is clearing out the hydrogen, so it's a buffer for the cell. And then two, it's an energy. That's a substrate for certain tissues. It can also get converted then into the liver via the Cori cycle to get turned into glucose. So there's many fates of lactate. And it's not bad. It's a good product that's being produced to help the cell out. It's a buffer for the cell. It says during times this is the original thought of why lactic acid was accumulating. They thought it was during times of ischemia, so low oxygen delivery to the muscle. Not a frequent occurrence in exercise, especially by normal, healthy individuals. We never really, with healthy individuals, get below critical level for oxygen. And so they thought there's low oxygen, so therefore the glycolysis system is working super high because of low oxygen. Nope, that's not it. It says when the hydrogen shuttles, glycerol phosphate are blocked. That's right here. This should be not a frequent occurrence, should be right up here. Okay, the hydrogen shuttles never get blocked at all. If they do, the cell's probably going to be damaged and probably could be something, unless you're poisoned or something like that, these shuttles will not stop working. So that's not going to be it. Number 3 is when pyruvate production rate exceeds the rate of oxidation of the muscle cell. That's the most common cause. Okay, so the most common cause, and I apologize for the slide right here, but the most common cause should be underneath 3. And what this is saying is that lactase formed because the pyruvate production, so as the rate of glycolysis, is far greater than the rate of oxidation of the muscle cell. So glycolysis is working at too fast a rate for the oxidative system to handle. So therefore, what's happening, it is then you got to get the cell going. You got to make sure to get the river flowing. So therefore, clear out the pyruvate. And you do that by converting into lactate, getting out of the cell to allow for the river to keep going. Nice flowing, nice and easy. Okay, so that's the main reason why lactase being formed is pyruvate production exceeds the oxidative rate. So the rate of glycolysis exceeds the rate of the oxidative rate. During exercise, the buildup of lactic acid indicates that the rate of ATP hydrolysis is high and will soon exceed the rate of ATP resynthesis. Hence, lactic acid buildup is an indicator of fatigue, not the primary cause of fatigue. So lactase not the cause of fatigue. Remember that. It's an indicator. It's associated with fatigue. Reason being is because as you're fatiguing, what's usually happening? You're usually exercising at a high level, at a high intensity. So you're tiring out from that, but byproduct of high intensity exercise is that, glycolysis starts working super fast. What happens then? Lactate gets formed. Always remember that. Remember that relationship. The harder the intensity, the greater lactate. That is, the higher the rate of glycolysis, the greater the increase in lactate formation. I will ask about that relationship throughout most of the unit. That is lactate and high intensity exercise. The harder you work, the more lactate is being formed. Why? Because just the rate of glycolysis is going up. As that lactic acid builds up, then the rate of glycolysis will slow down the rate of glycolysis. So that's why you tire out during high intensities, because, well, not lactase causing you the direct cause of slowing down. It's just you're slowing down because the cells become overwhelmed from many different products being built up. And so everything will start to slow down, so you clear everything out and get everything back working to normal. It says even at high intensity work, the pressure of oxygen level in the muscle does not fall below the critical pressure of oxygen level in the tissue. It says tissue oxygen level below which the muscle is oxygen starved. You can see the solid line, that's our level of oxygen as we start to train. And then you got the critical level at the very bottom. Well, this is just saying is that lactate, as lactate goes up, there's the lactate threshold, which we'll get into here in the next lecture. But basically, I'll just bring it up right now. Lactate threshold is the point which the body becomes overwhelmingly glycolytic, anaerobic. You can see lactate being steady, and then it starts to increase at exponential rate. That's the lactate threshold. That's where the body stops being aerobic and starts becoming primarily anaerobic. Well, on here, the pressure of oxygen within the tissue never gets below the critical level. So therefore, if it doesn't get below a critical level, then it can't be a factor into why we produce lactate. So it goes back to then, well, at the very bottom, percent of VO2. That's the reason why lactate is getting produced. It's because percent of VO2, as it increases, lactate formation increases as well. So there's the reason why we form lactase, because we start to train at a higher level, a higher rate, we become more glycolytic, therefore we produce more lactate. Okay, and this is just overall basically what we covered. Okay, you got the rate limiting enzymes at the top, so make sure you know those ones for each of the units, sections I should say, of metabolism. And he says control bioenergetics. Bioenergetics is the study of science, the branch of science that deals with just energy formation. Okay, bioenergetics is a part of metabolism. Metabolism, I've already said this, the overall reactions. Bioenergetics is a part of metabolism. Okay, metabolism is all the endergonic and exergonic reactions all occurring at once, looking at all collectively as a whole in the cell. Bioenergetics is dealing with just the production of basically ATP, to keep it simplified. Okay, so just kind of know, just go through all these parts. You can see all the different substrates coming in, where they go in at, and kind of know how they go in, and so forth for studying for this unit test. So that concludes with the energy systems. So to kind of recap, remember we have the three systems: the immediate, made up of the ATP PCR and the myokinase system, glycolysis, and then the oxidative system. Make sure you know the pros and cons of each. That is, like the median glycolytic system, they're on site. A disadvantage of the oxidative system is off site. Okay, and just know the advantages, disadvantages, and overall just what makes up these systems. Okay, the substrate for each one, the enzymes for all of them. You got creatine phosphate for the first one, glycolysis and glucose for the glycolytic system. Oxidative system can use protein, fats, and carbohydrates. Remember what you get from all the systems. That is, from the immediate system, basically just ATP. Glycolysis is to break down glucose into pyruvate, 2 to 3 ATP, and then 2 NADHs. Okay, Krebs cycle, 3 NADH, an FADH, and ATP. ETS, purpose, give us water and ATP. Okay, so if you just keep all those simplified and just know the basics of each one, just know what you get from all of them, the substrate, the major enzymes, and the overall just the process of these systems, and that will help you to understand getting into then the next unit, which will be looking at what fuel sources use at the right time. Okay, and so don't try and overdo it. Just keep it, like I said, simplify, keep it to the point, and just don't steer away too far from what we talked about. Know that with these systems that ADP is a big control of it all. So if you have too much ADP, then it will start, and you need ATP, everything will ramp up to give you the ATP. If you have low ADP and high ATP, then everything will slow down then to slow the systems down. Okay, know what happens if you run out of NAD. What happens if you run out of glucose, creatine phosphate, and those matters right there. That'll help you understand everything. Like I said, keep it simplified. Don't try and overdo it, and it'll just help you out to keep from steering too far off and getting yourself confused with trying to understand these systems and why the purpose of how they're working.